Thank you, Andy. Thank you, Bill. It's a great pleasure to be here, to meet old friends, to meet new ones, and to talk about a subject I really care very much about. Okay, quantitative biology. Um, and I thank Science Foundation for the interest in listening to this topic, which well, I've invested all my life in this, and I feel it's a very important one. So I want to start maybe with a historical perspective because we are here partly in this room because well, there's all this talk about the how 21st century is the century of biology, how we're in the middle of a biology revolution and all that. Right? So I want to start with just thinking about the sort of the scientific revolution we have come through. Um, so let's start with the 19th century. And so if you think about the 19th century, you think about steam engines, chimneys, assembly lines, and all that, right? And this is going on, uh, uh, the revolution is going on through today for 150 years. So if you think about what really underlies all of this output of this industrial revolution, we can point to just one thing. There's one trick of nature that mankind has figured out, right? And that's the trick of converting heat to work in a real controlled way. So that's the trick. But so for this to really develop into what it is, there's a enabling technology, right, which is a steam engine and steel, and the scientific foundation, thermodynamics. So if we go down to advanced by a century to the 20th century, so we lived through that revolution. We have transistors, VLSI, Wi-Fi, and all of that. So there's another trick that we have figured out about nature, and that enabled all this to happen. And this trick, of course, is figuring out basically figure out a way to describe and control electrons. And so the scientific foundation associated with that, of course, is quantum mechanics and this enabling technologies, integrated circuit, material processing, and all that. So each of these took a long time to develop, right? 100 years to develop to what it is today. So now we're in the 21st century, and okay, so we are, whoops, what happened? We are, well, this is something happened to. I don't. Okay, so the over here is supposed to be a list of all the wonderful things that biological revolution is supposed to deliver: rational drug design, bioremediation, saving the environment, and all that. And the so I really do not know what would be here that would be at the level of this type of really fundamental insight in the nature that we'll figure it out. But I do believe that if this bio-revolution is going to realize its potential as people have advertised, then I don't think sort of just stepwise optimization of something that's known is enough, and then there really needs to be some new insight about nature that's underlying that can be realized and developed. Okay, and so just like 100 years ago, 200 years ago, we wouldn't know the term thermodynamics. People just working through things. Probably, so right now we wouldn't know what's the problem that we're facing with, right? That will eventually understand and give us the breakthrough. But then, so if you think about in terms of the scientific foundation and technology, I think perhaps think DNA sequencing and synthesis is probably going to be part of the enabling technology. The new technology will appear. And in terms of scientific foundation, do not know whether we will know the right words yet to describe whatever the subject we're dealing with. It'll probably take a long time. But then of the type of things we've been talking about in biology, then quantitative system biology probably come closest to the type of thing that we need to understand about nature. But the main the thing I want to convey through this slide is that revolution takes time, right? And there really need to be fundamental work done, some new understanding of nature to realize exponential power. All right. So what is quantitative biology? Or QBio for short. So this term QBio, as far as I know, I think first came to our community in the context of this preprint archive that Paul Ginsberg and I put together. So this is was at the Los Alamos preprint archive for various mathematical. No, no, quantitative biology, so QBio, yeah. So the QBio is sort of the word that caught on. Yes, absolutely. Quantitative biology was 50 years ago. People were doing quantitative biology. And but then this QBio term sort of really caught on, and I noticed that Simon Foundation, UC San Diego, somehow singled out for supporting this. So then this QBio series of conferences have really taken off, and so there's a summer conference, there's a winter conference, and then there's quantitative biology communities and so forth. So various places spring up in the last 10 years. So let's say at least a revival of quantitative biology in the recent 10 years. But what is it? Well, different people have different, of course, ideas about what it is. So I just explain my view. So I don't think quantitative biology is merely about biology and the numbers, right? And it's not biology-inspired physics or mathematics, or just the application of mathematics and physics. Now, there could be all ingredients of those, and there's nothing wrong with them. But I think ultimately quantitative biology is about biology, right? So to learn, so for different disciplines to learn about interesting things in biology that we couldn't encounter elsewhere, and about discovering the principles of living systems. And quantitative biology uses approach that emphasizes on numbers as a crutch to gain conceptual understanding. Why quantitative biologists? Well, despite the fact that many biologists are quantitative phobic, math phobic, biology itself is quantitative, right? To a very good approximation, we have 5 fingers on each of our hand, and for very good accuracy, right? And and we normally have 2 copies of genes, and if you get 3 copies, you get seriously worried, right? So these are quantitative numbers. And if you're going to make any predictions beyond whether something is live or dead, you need to give a quantitative aspect, a social quantitative aspect to the predictions, right? So that prediction could be falsifiable. And then finally, if we're going to eventually realize the dream that's advertised to have do the design manipulation and inform interventions, as in biomedicine, in personalized medicine, and so forth, you need to know what to change, not just knock things off or get rid of things, but how much to change some things to achieve certain effects. Oh, the other reason the biology is quantitative. So biology is quantitative, but then so how should we promote quantitative biology? Well, we could wait for spontaneous mutation to happen to people like Eric Sijer to get them interested in biology. Or we could try to systematically try to cultivate an environment to allow young people to get into be bilingual and work on both sides of quantitative and the life sciences, right? So at UCSD, 2 years ago, we managed to convince our administration to commit to a program, a quantitative biology initiative, that would hire 15 to 20 people in a 3-year span to work at the interface, right? Now, just happened to notice that many of the people we hired, I mean, these are decisions made by, by engineers, biologists, and so forth. Many of the people we hire, the majority are actually have the phenotype or genotype of people that get a PhD in theoretical physics and then move on to do biology, either very closely to experiment or actually doing experiments themselves. Okay, I guess I can speculate offline why this happens to be a group that was selected. But let me move on to talk about just look more closely at the theory and the biology interface. So we, of course, there has to be reason for both to attract to each other to have a win-win situation for the two to come together, right? Uh, so to do this, I want to make another analogy. Okay, so suppose you are a Newtonian physicist and a Newtonian, there's a Newtonian physicist. Okay, and one day you come back home, riding in your horse carriage, and you see a automobile landing in your backyard. Right? What can you learn from this automobile? Well, you could say, well, I can apply it to calculus. I can figure out acceleration and stuff, and figure out how the car controls acceleration, figure out how the suspension works, and maybe design how the car you should use the car and so forth. You could notice some phenomena that's really brought out by having a fast car. Look at the hydrodynamic flow, and maybe you will write down Navier's equations a couple of decades before Navier did. Get inspired by a phenomena and write down some interesting new mathematics. And or you can use it as a chance to discover new science. And so, what are examples of a new science you can discover with this car? Well, you can open up the dashboard and discover there are these chips in there. And if you're curious about how they work, maybe that will lead you to quantum mechanics. You can open up the hood and try to figure out how a heat engine works, and then you'll be 100 to 100 years ahead. You'll find out thermodynamics. Right. So, of course, what we have is instead of the car, we have these biological cells that are sort of very clever in many ways. We'll look at it. And so how can theoretical physicists and the mathematicians in general, uh, benefit or contribute to understanding of these guys? So from what I have noticed, also the interaction seems to be basically clustered around these three kind of in these three kind of ways. And so I'm going to describe for you sort of my own experiences, pick one in each of these areas. Okay, so I'm going to give you a little bit of a short talk on each of these three areas. One is in molecules, where our contribution is mostly in methods to try to understand something about, say something about molecules. One is in the area of pattern formation, okay, and one is in the areas of phenomenological laws. And actually, another thing is that the different styles of how theorists interact with biologists, and it just so happened that in each of these examples there is a different style of interaction, and I'll illustrate them as we go through these problems. Okay, so let's first talk about this biomolecule problems. So this is about meta-analysis. So as you all know, it is a very difficult problem to predict the structure of a protein from a primary sequence of amino acid. Okay, this has been an outstanding problem, and we don't have anything new to contribute to that. This has been a grand challenge problems for biology for a long time. But what we have realized is that maybe by combining using the information that many similar sequences are known, the sequence proteins that have similar structures, maybe can exploit this information to say something about what the three-dimensional conformation of proteins might look like. Okay, so these sequence information brought forth by the lots of genome projects, and in particular nowadays there are thousands of bacterial genomes that have been sequenced, and then within each genome, the certain proteins have a highly amplified, have many homologous. In one genome, there could be 10, 20, 30 sort of similar type of proteins. Okay, so then so the main premise of this approach is that if there is so there's the constraint in the structures, for example, these two residues touch each other in a three-dimensional conformation, induces correlation between the sequence. So that if these touch each other and one residue change, then the other residue also have to make a compensatory change to accommodate the structure. All right. So, so this can be picked out by looking at two columns of so these are multiple sequences that are aligned together for the same type of a protein. So the very established way to do this kind of alignments. And so you can see that in these cases, when there's a certain residue, S is here, there's always a P here, but one makes a change, then the other side is also this compensatory change, right? So this kind of a this correlation can be picked out by counting the the frequency at each residue and noticing that the joint the joint distribution of the frequency at the two sides is different from the product of a single body correlation. So this depends on the detailed amino acid composition. But then if you just want to boil it down to one number to see how correlated these sequences are, you can use a number of measures, such as a mutual information that captures how much correlation is between two sides, say I and a J, along the backbone of a sequence. Okay, so now to infer the interaction, the one problem with this mutual correlation is that correlation does not directly imply interaction, right? So I hardly interact with anybody in this room, but we all come here together in the same auditorium, and we're Andy Millis is responsible for getting us together. So we have a direct interactions through Simon's Foundation. All right, so then what the task is really to, from the mutual, the correlation to extract the direct interaction between pairs of residue. And that could be done in a number of ways. And so particular, so what we know is we have a distribution of so we have n sequences, and we have a many-body distribution function for which only the two-body distribution is known. And so in a maximum entropy approach, then the best way to describe this many-body distribution is described by the system, which is involved just pairwise interactions. Okay, in statistical mechanics, we call it a Potts model, the 20-state Potts model for the 20 amino acids. So the task is, given the observation, which is a correlation, how to deduce the interaction. And this is a, well, no inference problems. Okay, so this inverse correlation, it's a challenging computational problem to do this inversion. And we work together with Martin White, who was in France now, and develops a way to do this inference. And later on, Chris Sander joined us, and we developed a more simpler way to do this inference. So we call this analysis to retrieve the direct interaction analysis. Yes? Yes? You don't know if it's, you just do so the data you have is only enough to give you basically a the pairwise correlation. Now, nowadays, maybe you can start to get some information about 3-point correlation. Just yeah.
My question is, how do I know that, as it were, the argument of your exponential is just an epsilon ij of aiij, that there isn't a three-body term or four-body?
So there could certainly be two-body and a three-body term, but then to fix those, you need to have three-body information of three-body correlation. And those cannot, at the moment, still not reliably estimated from the existing data. So you do the best you can with the
But then I guess, why is it good to know this if it's uncertain because I don't know the 3 and 4 and higher?
Yes, okay, so this is a practical problem of, given what reliable data you have, you do the best you can. Right, so this is certainly you need to have a two-body interaction. It's a lot more than one-body interaction. Yeah. Right. And okay, so this interaction matrix still involves all 20 amino acid by 20 amino acid, but then again, but then what we want to know is just some sense of a correlation, and this can be then we develop a measure called direct information, which is basically modeled along mutual information, except that the correlation is computed according to the model. Okay, so then there is a number that summarizes according to this model the interaction between two sites. Okay, and so this was used to scan over uh, all the available protein families at that time that have the sequences and also the structure, just to see how good this is. And you see in this case that so this is like the top 20 or 30 this correlation according to this model, and the red ones are the ones that agree with the actual structure. So this one's rather good. And in comparison, if you just use the correlation measure, the mutual information, then you get lots of unreliable prediction. Okay, and then this was so this was for one anecdotal examples, and we did for this all this 130 protein families, and you see that the number of true positive, the red bonds, percentage of the red bonds are much higher compared to this mutual correlation. Okay, I want to just walk you through two aspects. First, if you look at, say, the top 30 contexts, which are very good, they're still not perfect. So what's wrong with them? Well, so here is an example of a protein. So out of the top 20, 17 are good predictions in that the predicted, good, ones predicted with a strong interaction are sitting basically next to each other in three-dimensional structure. These three are false positives. They're far apart in the protein structure. But if you realize that these proteins actually, in reality, they work together as a multimers. In this case, the 7 protein comes together as 7-mers. And you put these contexts on top of 7-mers, you see that these 3 pairs becomes a good context. They're really sitting next to each other in a multimer context. Okay, and this is actually we find that half of the so-called false positive are just basically this type of things, and the others you can also nail down. So very biologically meaningful things. So the top 30 also contexts we really have a very strong confidence about. So what does the method produce? Okay, and you can use these reliable contexts to assemble multimeric proteins if you know the structure of the single proteins. All right. So another way to look at this is so if you can accept a predicted value of a 70%, right? Then you see with this direct coupling analysis method, you can get good information up to you can basically believe in your top 100 predictions. That's a lot of numbers of predicted contexts. Okay. Uh, So if you look at what they are, so here is a contact map. The gray is showing the true contacts that's made between residue I and J. And the red are the ones that's the true positive from this method. You see that the red are basically sprinkled quite uniformly across the Bay Area. So each of the regions of a context, this method is picking out context. Whereas in comparisons and with mutual information, mostly getting just very clustered, missing most of the context. All right, and all right. So basically, what it points to is that this DCA is capturing a lot of context that's far along in the backbone distance. Okay, and these are actually the most difficult contexts to get for a proteins.
Gary,
Yes.
how do you distinguish in this approach interaction which is of a physical origin, that they just couple to each other because they just match,
Yes.
or evolutionarily that it's simply a group of proteins, or most proteins belong to this family, and they just carry this along? How do you distinguish historical from physical?
So this is, of course, an evolutionary approach. So it's assuming that evolution has long enough time to act, so you're kind of in a steady state where it has explored all the phase space and it's accepting all the pairs that could give you this structure. Right? So basically, the underlying picture is that you have some real physical interaction in mind, but it's not specific. Only one residue can be with the other residue, and the evolution has enough time to sample all from this distribution.
It is mostly evolutionary.
Yeah.
Okay.
Okay. All right. So then it occurs to us, since there's so many contexts, okay, especially long distance contexts, maybe we can use it to say something about protein folding, okay, without knowing anything about the structure. So then we worked together with Jose Onochik before he moved to Rice University. It was still neighbor down the corridor. And so Jose's group used this what's called a Go model. That is a model where if in the final structure, if you know two residues next to each other, you just put a van der Waals spring between them. And he used this kind of model to study the kinetics of protein folding and so forth. But we said, well, why don't we just use the same model, but put the van der Waals spring only across the residues that's predicted by this method to be a true contact. Okay, and just let this system evolve. And the idea is that if there's enough coherence, then the ones that's far apart will not contribute. Even if we make a wrong prediction, if they're far apart, van der Waals interaction falls off quickly, then it will not make a difference. Okay, and the methods works reasonably well. So like in this protein, which is about 100 amino acids long, you get a near atomic resolution for the kind of a structure that's obtained. Okay, so what I should say is that what is inputted into is a long distance. Okay, long distance interaction from this statistical method, and then the short distance you need to incorporate the secondary structure. Okay, but because there's no way that the system will know to make this right-handed helix. I mean, if you do not tell it the local structure. All right. So, but there are many methods that do a very good job of predicting secondary structures. So we just took any arbitrary method and put it in. Okay. Yes.
Geometry. So you're predicting the distance between
No, so what you tell her is that, okay, I think from the statistical analysis, I think residue 3 and residue 27 are interacting. Okay, so then you put a van der Waals, you put a van der Waals spring across residue 3 and the 27. That's right. So the other ones just don't do anything. Okay, it's only the ones that's picked out do. Okay, so about 30. Yeah. So 30% are wrong, but there are enough good ones. Okay, to make this collapse. All right, and so this was done for, we examined a dozen or so structures, and some are better than others, but more or less in the range of a 2 to 4.5, 5 angstrom, this kind of residue. Okay, but realize this is starting from nothing, just starting from the sequence and put it in. Okay, and then now people that know something about Go model, they're always afraid that, oh, Go model, you're putting the answer into the system, even though we are not in this case. Okay, but here is really something that shows the power of these methods. So we look at a number of proteins that have two states, two conformations. All right, and so again, you do the same analysis. You put the van der Waals spring across these, and you just run them. And then the trajectories, the conformation can be clustered into 3 groups. One is within 0.9 angstrom of the open conformation. One is within 1.1 angstrom of the closed conformation. And then there's the intermediate state. Okay, so in this particular case, the intermediate state actually has been captured in the crystal by they provided some chemicals in there to lock in the intermediate state. But this method just has 3 clusters, and these happens to be the 3. And we didn't put in any of these states into it. In fact, very difficult, even if you know the final structure, try to get some simulation to capture the intermediate states. But this one just naturally. Okay, so again, we did this for a number of systems. Okay, so to summarize this type of work, so we'll say that this statistical analysis method, direct coupling analysis, provides many reliable long-range contexts, and then they can be used to, in the cases we've looked at, to predict the structure of proteins. Now, people say, well, why don't you then, how good is this method? Why don't you go to test some of the unknown proteins as these protein structure prediction contests and so forth, okay? Well, there's a problem. Okay, the problem is that the bottleneck of this method, it consumes lots of statistics. You need roughly 1,000 sequences that's not too similar. If they're too similar, then that does not carry information. Okay, roughly you need 1,000 sequences that's say less than 80% similar. The very few proteins in the structure that have 1,000 sequences and do not have a structure. Most of the proteins, when if people bother to have kind of that many sequences, they've gotten the structures already. Okay, so I think to me really I think the outcome of our work is to demonstrate to people that if there are a lot of sequences, there's a chance that this method could tell us something about structure, and really there's a need to do sequencing for if you're interested in this. The need to do sequencing for homologous genomes. So, for example, at the moment, bacteria sequences there are thousands of genomes. Okay, but easily with today's sequencing technology, one can try to sequence, say, 1,000 yeast species, and then use use this information to get proteins that's not, but eukaryote-like, that's of importance to to human. Yeah.
This method would not allow you to think about proteins which have unnatural forms, which were never
So
realized in nature. Chao Teng has tried to extend it, I think. So
yes.
is it possible to extend?
Well, so I mean, this method is based on sequence that exists in nature. So if you design something that doesn't exist in nature, yeah, there will be difficult. But they've actually Jose's postdoc have used it to actually to look at protein that form knots, and they could say something about even those proteins. Okay. All right. But before I move on, I want to bring out, to make one commentary about the interface of the theory with biology. I think as anybody who have experience of theorists going to biology, so one of the biggest, most important thing about doing useful theory to biology is the need to get hold of a reliable data sets. Okay. When I met Martin White 5-6 years ago, he was working using the same type of inference method to deduce genetic networks from microarray expression data. Okay, and I think the most useful thing I did in this part of me was basically I told him life is short and work on something where the data is reliable. So for people have experience with microarray, let's say, or many other sorts of data. Let's say the quality of data are heterogeneous at best. And when a theorist that doesn't know about what the underlying biology is just grabbing data from internet, there's a lot of danger of what can come out of come out of this analysis. Okay, so for genome sequences, for protein sequences, it so happens the genome sequences are the most reliable source of data, and we can just interact with the data in this way. But what about other type of problems where you really need to steer your way carefully to get the reliable data sets? So there are two approaches to do this, right? I liken them to hunting and farming. So the hunting style is so every now and then there's a new method come up, or there's a new biologist that you happen to be able to that's in your vicinity that you have a good chemistry working with, and you work with them and you get a hold of data, you do analysis. Next month, some other thing come up and you do something else. Okay, so you could contribute, but then it's difficult to sort of have a program, a coherent program you can control. Today you eat rabbit, tomorrow you eat deer. They're all both very tasty, but kind of you don't have your choice. Maybe limited, unless you're very good at reaching out and interacting with biologists. The other style is a farming style, where you choose what problem you want to do, and you cultivate the data that's needed to solve your problem. Right. So I'm going to go on to, of course, there's a whole spectrum in between, right? So then I kind of I'm not a very good hunter, and then I try to sort of live in a farming kind of style. But the difficulty, the problem with the farming style is you eat vegetable, right? You eat what you plant, but you don't get as much of a variety, right? Okay. So then let me move on to talk about another problem I have to do with spatial temporal patterns. So pattern formation in biology is everywhere in biology, and has fascinated many physicists, mathematicians alike to think about things. There are basically, I could say, two ideas in the formation of patterns. One is this idea that there is an external coordinate system that's laid out by some chemicals called morphogen. Okay, it's produced at the head, and say decayed away, so that lay down some gradient. And so if you have a system that responds one way if this concentration of the small function is above some level and another way below some level, well, then you can define regions. Okay. And for example, different patterns can arise in this way. Okay, but this type of patterns, it requires some external agent to lay down some axes, some polarities. There's another type of approach, reaction diffusion system, that's gets aimed at the self-organized formation of patterns. Okay, so typically these type of a system have a number of interacting diffusing agents, and they interact and give rise to patterns. So the most famous one was the one that's analyzed by Turing, where you have a two species activator and a repressor, activator activate itself, and activate the repressor, 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 activator. And then the condition that this activator either decays fast or diffuses slowly, then we can get into this situation. So commonly described as a short-range activation and long-range inhibition, and so patterns can arise when over short-range activator overwhelms the repressor. So in this case, a two-spot or stripe can arise. All right, and then by playing with the details of these synthesis function and the parameters, one can get a uniform system to generate different type of patterns. Okay. So I've always thought that something important is missing, an important ingredient is missing in this type of approaches to thinking, and the one that I was looking at was the fact that in many developmental systems, the physical expansion of the embryo or the system is important ingredient, whereas many of these systems will think about in the context of a closed system, a fixed spatial domain. Okay, and so I've wondered about how to how to think about the incorporating the ingredients of the physical growth and the motility and also signaling. These, I think, the three key elements in making patterns. And then in a opportunity presented itself. It is, I've been a regular visitor of the University of Hong Kong for various reasons. And at some point, the students there want to do a synthetic biology project. Okay, to synthetic biology is a very hard thing, so get many students are involved. And then so I first tried to persuade them not to do it, but there's just no way that they're going to do it. Okay, so I said, well, maybe we can use it to learn something. Let's try to throw these elements together and see what kind of patterns can be created. So in the laboratory of J.D. Huang, who is himself a developmental biologist interested in this kind of more conceptual questions, we decided to develop some bacteria that would have the would try to realize these three elements in the simplest way. That is, we have a bacteria that will grow, that will move around, but then it will respond to signaling as a function of the state of the bacteria, and the simplest state of the bacteria is the density of the bacteria. Right? So we generated a system where if the density of the bacteria is high, then they will not move around. Whereas the density of bacteria is low, they will just swim around freely. Okay. All right. So what happened to such a system? So here is a movie. So we made these bacteria and spot them in the middle of the plate. Okay. And okay. So then after this is in hours, after a number of hours, then. Okay, so the bacteria expands and form these stripes. Okay. So it's whereas a wild-type bacteria will just expand and do nothing. Okay, so So there's some instability going on. Why this is the case? So you can understand the source of the instability quite easily. So imagine if you have a region with a high density bacteria, a region with low density, and then the low density guys will move around. But if they happen to run into a region with high density, then they'll stop moving. Okay, and so then this means that the high density will suck away the neighboring region. This is like absorbing boundary condition, and you have a separation of the high and the low density. Okay, but why periodic stripes? All right. So to do that, just doing simple linear thinking is not enough, and we need to write down some equations. And so in this system is simple enough because we control what it is. So there's a signaling molecule, okay, that we put into this is E. coli cells. Put into E. coli cells. So the synthesis of this signaling molecule is proportional to density, and it decays rapidly and it diffuses around. Okay, it's density dependent synthesis. What happened to the cell? Rho is the density of the cells. Well, they consume nutrients and they grow exponentially, and they move around. Okay, there's a motility coefficient called mu. So the way bacteria move around, so they're too big to move around by Brownian motion. So what they do is they will swim for a while, maybe a second, half a second, and then they will tumble, pick another direction, and then make a run and so forth. So they trace out a random walk. All right. Now, what we did in the system is we engineered a system to control the length of this run. Okay, so we engineer a system where, according to this signal molecule, which is generated proportional to density, if the signal density is high, then the run length becomes smaller and smaller. Okay, so if this density is high enough, then they just basically go around. They only tumble, they don't run at all. All right. So this is actually data to show that we indeed constructed what we thought we did. So this is measuring the diffusion coefficient. This is in uniform density, and as the density is high enough, then the diffusion coefficient drops by order of magnitude. Okay. So then this type of behavior is described by this kind of a term, Laplacian of something. Okay, note that this is not it's not what physicists call diffusion, because the diffusion will be something like this, and the system is not. You know this is not diffusion because if you add a constant density to the cell, diffusion will not respond at all. But this system is completely different depending on the absolute level of density. Okay. But anyway, so we model this system by a simple function where this motility coefficient drops by a lot as the density exceeds some threshold. Finally, the nutrient diffuses around and is consumed by the bacterial cells. Okay, so in this system, the only non-trivial interaction, there's only one non-trivial interaction, and that's in this motility. Everything else is simple. Okay, so this system is sufficient to generate these patterns. Okay, so let me just show you a simulation now of just these three simple equations. The blue line is the cell density, red is the signaling molecules that excreting, and the green is the nutrient. So as I turn on the system, then the cell density is going to accumulate, nutrient is going to go down, and the signaling molecule is going to also follow the blue line. It's going to increase. This line here is the threshold concentration of signaling molecules above which the cell motion stops. Okay, so everything is just going to expand until the red line hits this dashed line, and then the blue line is going to do something different, right? So, okay, cells start growing, and the red line is going to hit the black line. Okay, now you see a buckle develops. Okay, there's this thing that moves around. Okay, and then at some point it repeats it again. And it repeats again. Okay, so the system generates lays down a periodic patterns behind it, right? So if you look more closely at the system, first thing you notice is that the front actually is always below this line. So the front always propagates just like a wild type. It doesn't know anything about the interaction. But then the the lagging region eventually stops growing, right? And it sucks the neighboring region by this instability. So that digs a hole into the profile. And so if the front keep on going and the region behind just stays put, then that's going to generate a gap and a new thing is created. Okay, just from the propagation of the front and a simple instability, it's able to regenerate. This structure is able to regenerate itself. Okay, so we have a autonomous pacemaker, right? Periodic without any clock, and here you see that the expanding spatial domain plays a crucial role in the system. All right, so we can do some analysis, actually solve for, you can solve for a steady state moving solution and look at the instability to get a phase diagram. And the phase diagram basically depends on, the key parameter is how fast the cells are moving in the resting, in the low density phase, okay? So, so in terms of a parameter, so there's some regions where the system can form many stripes, few stripes, and no stripe, and and one can dial through these regions by changing the diffusion in the basal diffusion level of the cells. Well, so we designed a system to implement exactly that. Okay, so never mind the detail of what we designed, but you can see that so the system we put in, we can adjust the peak level of the control of this diffusion as a function of density, and we can this peak level is also controllable by a chemical that we put into the medium. All right. So, and this is the result of this test. So the same bacteria, just putting different chemical concentration, and they create a different number of a stripe like this. Okay. So now this is, of course, the type of a picture biologists have in, this, in kind of the textbook pictures. We're changing some chemical, then patterns can change. But it's actually very difficult to demonstrate this in a real living system, because once you change one, it's very difficult to change just one component. Often when you do something, you change many things. Right? So in this kind of a synthetic system, it's possible to make simple changes. Okay, so the lesson here is that for this problem is that motility control, the kind of a control that we normally do not often encounter in physical system, is a very succinct way to generate patterns. And this type of control is really everywhere in environmental biology. Okay, the control of either the movement of the cell or the movement of the molecules as a very heavily used actually by cells. So this is something that when we think about developmental patterns, some ingredients to keep in mind. Comment about the theory biology interface again. A crucial aspect in the study is really the quantitative characterization of the system. So I told you this was a result of a synthetic biology project. So I was there for a number of months, and so get people to make these things. And a half a year later, I came back, and so they made something that showed a little blob. Okay, and but that's fine. That could be what the system is, or it could be that what they thought they had was not what they actually had. Okay, so then we went through great trouble measuring everything. Okay, so actually in this paper there are 4 or 5 sort of measurement methods that you do not. I mean, it's not just sort of off-the-shelf method, but the students have to develop each of the methods. So measures as a function of density, the diffusion of the cells, the mRNA levels, and so forth, or in the context of this agar gel. Right? And so the students are very capable going and do the characterization, but until they do the diagnosis, they realize what system they actually had, and then they made the ones that eventually made the ones that was intended. Okay, and then this sort of a behavior came out. Okay, so I would say this is a model of a demanding collaborator. Right, so I was the theorist in this project. The experiment was carrying on in another lab, but I had to be very demanding on everything. So, okay, now this kind of interaction is not always easy to come about, and in fact, I have difficulty convincing my own colleagues in UCSD to do this kind of things. You have to really push the limit. I was able to to get my demand in this case because University of Hong Kong, the chancellor actually asked me to go there to help develop quantitative biology and gave me some fund, so I was actually throwing out the resources to get this kind of a this kind of a project done. Okay, but that's one difficulty that until the experimentalists see the value of the quantitation, this is why we need to invest all these efforts. Okay, so let me then move on, boy, to talk about the last subject, which is phenomenological characterization, physiology. And this is where I think in the portfolio of what we do is closest to seeing something new in biology. All right. So let me go back to you may ask immediately that, well, why should there be something new? I mean, there could be just details of interactions. Okay. So if you go back to this historical development, so we can usually associate a reason, right, for when there needs to be there could be something new. So for quantum mechanics, it's clear ultimately that in retrospect at least, the scales are very different. So we have no reason to demand what happens in our scale of tennis ball to apply to electrons. For thermodynamics, of course, large numbers that gave something new. Well, what about here for biology? Well, one thing that's very obvious is that it's just so heterogeneous. Many different systems. Every molecule is different, right? So this was a really old diagram of a genetic network where you have a number of genes interacting with other genes with a sign that's either activating or repressing. Now, if you're digging into each of these type of promoter region, you have interaction with different type of proteins with each other, and so each of these can depend on their state of phosphorylation and so forth. And when you put it all together, you're getting microscopic number of parameters. Okay, so we as physicists, we just don't have any experience of dealing with this type of a system where okay, so yes, we believe ultimately this is there's no magic, everything is explained by quantum mechanics or perhaps just biochemical chemical reactions. But then what do you do with such a thing, right? Okay, so so then we have taken on this problem using a phenomenological approach to look for simple behaviors, because obviously the biology cannot just work by kind of just let these parameters decide for themselves what's supposed to happen. Okay, so we look at a problem, a problem that was motivated by a study from 50 years ago, that the simplicity in behavior. Okay, so this is a problem of bacterial cell growth, just exponential growth. And so if you have a culture, a medium with a carbon and nitrogen and so forth, and you seed a few bacteria, and then the bacteria grow exponentially, and what's measured here, the optical density, which is a proxy for cell density. Okay, for a while until the medium's exhausted, and then the growth saturates. Okay, so in the exponential growth phase, it turned out that there are a lot of simplicity, the simple behaviors that one can point the fingers to. Okay, so at the moment, so this just described by exponential growth is described by simple rate, the exponential growth rate, lambda. Okay. So then people, since 50 years ago, they have done lots of studies taking cells in the middle of exponential growth and measuring simple things that they could get their hands on, RNA, proteins, and so forth, DNA. And they notice something that's quite striking. So here's some results for E. coli and its cousins. So the x-axis is the growth rate. So for each of these points, it's exponential growth. You feed the bacteria with some food, okay, and it's growing at some rate. And then you measure the total RNA in the culture, the total proteins in the culture, and you take the ratio, and you see there is a near linear dependence, okay, independent of the particular medium that's growing on, just on the growth rate. This is true not only for bacteria, but this is also for several fungus that's being studied. This is a protist that divides once every 24 hours, okay? So for a range of a growth rate, this type of behavior is seen. So what does this mean? RNA is a proxy for ribosomes because most of the RNAs, stable RNAs in the cells are basically ribosomal RNAs. protein is a proxy for cell volume, okay, because the density of the cells, cells made mostly of proteins, and the density of the cells is constant unless you change osmolarity. So this is saying that so what this ratio is saying is that the ribosome concentration is a linear function of a growth rate. Now, most microbiologists know that at fast growth rates, the ribosome content in a cell is high, but then probably not many realize that there's a very simple relation. Now, so who cares? Why? So what? This is a linear relation. Well, Oli Moeller, whose lab first discovered this simple dependence, had a very good understanding of the underlying process. Okay, she says, okay, so ribosome first of the ribosome is the agent that produces all of the proteins in the cell. All right. So suppose all the ribosomes are working efficiently, right? So in making proteins, then the total rate of protein synthesis is just the rate how fast ribosome work. Okay, so in exponential growth, the rate of a protein synthesis is just the growth rate times the total mass, and this is the rate of a gamma is the rate of how fast ribosome works, and this is the number of ribosome measured in the mass of ribosome. So if you set these to each other, then immediately you see the fraction of ribosomal protein should be a linear function of growth rate. Okay. And if you look at the numbers, if you look at the slope convert to real units, you find that it actually corresponds quite well to the known elongation rate of ribosome. Okay. There's an offset, but that could be explained away by saying so that you need to have a pool of free ribosome that's ready to engage in translation when called upon. All right. So, so that was the picture. But then a question is then, but how does the cell know how fast it should be growing? So you can give it a combination of a nutrient that it has no chance of seeing, in billion years, right? Some source of carbon, nitrogen, sulfur, and so forth, all right? And it kind of picks a rate and it grows at this rate, and all of these things are balanced. Right? So how is this possible? So then a while ago, we made up a model, a simple theory of develop a simple theory of bacterial growth that's based on lots of experiments that's reported in this paper, but I have no time to describe to you. The model is very simple conceptually. You provide the cells with the nutrients, and you need proteins to convert these nutrients into amino acid, and these amino acids then polymerize to make ribosome to make other proteins by ribosome. Okay. So this is just a simple linear system. And so we envision that in the proteome of a cell, there's a fraction of them, of the proteins are the ribosomal proteins. A fraction are these proteins that provide the nutrient. Okay, they each have their rate, kinetic rates of working. And then there are other proteins that's as far as this is concerned, that are fixed, that do not depend on the nutrients and the growth rates and such. Okay, so then one can write down a equation. We already know there's a linear relation between the ribosome fraction and the growth rate, and the experiments also suggest that there's now experiment that there's a linear relation between this nutrient protein that provides the nutrient flux and the growth rate. Okay, so then the task. Okay, so nu here has to do is how fast the protein processes a nutrient, and it's a property of the nutrient. So basically, then in the system, there's one more. So the PNR has to add up to about 50% of the cell. Okay. So given a nutrient, then the cell has a task of partitioning the ribosomal protein fractions so that all three equations are satisfied in this simple picture. Okay. So there's a simple analogy one can make to the system. This is a simple analogy of two resistors in a series. Okay, so in this analogy, the current flowing through the resistor is just growth rate. The voltage across it is these proteins. So you need proteins to push the flux. And so with this analogy, then you see these linear relations are just Ohm's law, linear relation between current and the voltage, right? And these coefficients are just the state variables, the conductances of the material. Okay, and then this is a voltage addition. All right, so we have a simple problem of you put some material with some conductance, that's the nutrients in this case, and you're pushing a flux through it, and you see how much protein you have to to allocate to push the flux through. All right, so we can write down the total current in terms of the total voltage across the system, which is the total amount of protein that's available to span on the cell growth. And so this is a linear system, so the inverse of the conductance is R, so we have this simple relation. This is actually quite an amazing relation if you think about it, because this is a relation between growth rate and this nu is a neutron, can be neutron concentration, a neutron property. And it's a Michaelis dependence between growth rate and a neutron. Okay? And so this was actually something that Jacques Monod discovered in his thesis in the 1940s. So he found out, as complicated as the cell is, when you look at the growth rate as a function of a substrate concentration, it follows a simple Michaelis relation, as if it's just a simple enzyme that's working. Okay? So this kind of just fall out of this simple model, and even the numbers sort of make sense because the coefficient that's sitting in front, that would be the Vmax of this enzyme, the maximum growth rate, is rather close to the 20 minutes per doubling type growth rate that we see for E. coli. This is just number just pop out. Okay, but of course this could be a coincidence. There's so many coincidences in biology, so we wanted to test this. So the way to test this, I mean, this is just a current voltage relation. What you need to test is a change voltage and see how current change, right? So what we decided to do is, well, we cannot mess with the cell, but what we can do is to force the cell to express a protein it doesn't need, okay? So this is actually, people do a lot of that in biotechnology. You force E. coli to make an insulin, right? And that's gonna hurt the cell, okay? But let's see if we can predict how much it hurts the cell. So according to this simple model, if you force the cell to express some other proteins, that's going to take away from the proteins that ribosome can direct it to synthesize these RNP proteins. Okay, then the only thing that happens is this maximum available fraction is reduced by whatever you force the cell to express, and then the growth rate should be reduced as a linear function of this forced expression. Okay, just a way to test Ohm's law. All right, so this is completely fixed relation. Every number is known, okay? And so the lines are just drawn according to the measured numbers and the green points on the data. So basically what it says is as you force the cell to express more and more proteins, in this case up to 30% of the entire proteome, so you force the cell to express lots of proteins, growth rate goes down, but in a simple linear fraction, as expected from this simple picture. So this sort of experiments were of course done in early on in the 90s, where people were worried about the effect of overproduction of proteins, and we were able to find data where people use the same medium, okay, and to look at the E. coli cells, and you see that for various kind of protein that's overexpressed, the different data from different lab collapse onto each other without any adjustable parameters. This is a real growth rate. These are the actual absolute fraction of the protein that's expressed in a cell. Okay, so there's a lot more to this, but I think but the lesson to be learned from here is that this kind of data, I would say, is beginning to approach the level of quality of the data that physicists are comfortable with dealing with, right? So we see that the simple behavior can come about despite the enormous complexity that's underlying the cells, right? And a quantitative predictive value is possible. In this case, 0 adjustable parameters. Right? And so obviously we're applying a coarse-grained phenomenological approach. Phenomenology is dirty words in biology, okay? But in this case, it can lead to predictive results, right? And certainly physiology is something that one is not paying enough attention to. Okay, this is my last slide. The work that was talked about here is deal with expressing heterologous proteins, talk about ribosomes, which could be special players. We have since found that even plain old regulated genes, many plain old regulated genes, also obey simple linear dependence when plotted with a growth rate. Okay, so this is a subject of a paper that was just recently appeared. So this is not only for a specialized system, but it's happening all over. Okay, and so I'm going to make an advertisement. So I'm going to give a talk tomorrow at Columbia Medical Center. It's a bit far up town on this project. So we use this type of phenomenological approach to study one of the most classic system in molecular biology, catabolism repression. Okay, and we derived significant different meanings, physiological meanings of what catabolism repression is and does compared to what's taught in biology, introductory biology textbooks. We're able to, using this approach led us to identify molecular links. So usually phenomenological theories think, oh, just kind of a coarse-grained quantities, but we're able to get molecular links on what interactions were missing, from 50 years of classical genetic studies, right? And also the high throughput studies, but we were able to capture to explain this kind of a simple macroscopic relations. This then leads to a new picture of a hierarchical carbon usage, mixed carbon, all the classical stuff about metabolism, bacterial physiology. So we have a new way of looking at this and a rather different picture, I would say. And also, we've released this picture of a protein partition, this R sector, P sector, and so forth. So far, we've sketched as a cartoon models. We have done quantitative proteomics and actually physically show what they are and can tell you how they change and what they are in what are in each component and so forth. Okay, so. Finally, I just leave you with this picture. Again, if 200 years ago, people knew, gas in this room is made of molecules, and they collide with each other and follow Newton's rule. Okay, what could you learn from that? And let's say you have technology to follow the trajectory of each individual particles. All right, could we discover ideal gas law from analyzing the trajectories? We can debate about yes or no, right? But I can tell you one thing I think you'll agree on. If the experiment were done, okay, with temperature, without knowing temperature, with temperatures fluctuating wildly, you're not going to discover anything by looking at analyzing this trajectory, right? So there are two roles I think theorists, physicists, mathematicians can play in this. One is to help analyze the trajectory and get information. The other is to tell the biologists to keep the temperature constant in the experiments. Okay. So thank you for your attention.